Okay, everybody, Dr. Rafich here from Vet Triage. We are continuing through this issue of JAVMA, May 1st, 2014, and we're going to be talking about ferrets. This study comes out of France. We are looking at the prevalence of deafness and association with coat variations in client-owned ferrets. The objective is to evaluate the prevalence of congenital sensorineural deafness and its association with phenotypic markers in client-owned ferrets. It's 152 healthy European pet ferrets. They use a test called the brainstem auditory evoked response test, or the BEAR test, during general anesthesia for the ferrets. The results found that 29 ferrets were affected by congenital sensorineural deafness. 7% were unilateral, 22% were bilateral. There was no association between this deafness and sex or Angora trait, but a strong association between deafness and white patterned coat or premature graying. All panda, American panda, and blaze ferrets were deaf. Conclusion: breeders should have a greater awareness and understanding of this defect to reduce its prevalence and to the overall benefit of the species. All right, introduction. Coat color-related sensorineural deafness has been described in numerous species, including cats, dogs, horses, mink, mice, guinea pigs, pigs, llamas, and alpacas. White or merle or dappled color coats are associated with deafness. Deafness is linked to failure of migration or maturation, premature death, or dysfunction of neural crest melanocytes in the inner ear in a region of the cochlea known as the stria vascularis. The BEAR test, or the brainstem auditory evoked response test, has been used to identify deafness in ferrets. This electrodiagnostic test provides information about the functional state of the peripheral and brainstem component of the auditory portion of the nervous system and can be defined as the electrical response of the auditory pathway to a series of auditory stimuli. This procedure can be easily performed in a clinical setting in ferrets as a screening test before adoption. BEAR test is an objective method for the diagnosis of congenital sensorineural deafness and can potentially be used in a program of screening and selecting breeding stock. So it might be obvious, but unlike people where you can play a sound in the ear and you can tell the person if you can hear there or not or whatever, you can't do that with animals. So you need to have them under anesthesia and provide electrodiagnostic tests like BEAR test to figure out if they can hear or not. So here's some of the coats. We have an albino ferret up top there with the red eyes. And you've got here at the bottom, let's see, A is chocolate, B is pastel, C is sable, and D is black. And then here's some other coat colors they're going to be testing right there. You can see those are the white patterns there. So A is American panda. A is panda, B is American panda, C is blaze, D is silver, and then E is DEW, which is, hold on one second, dark-eyed white. So DEW right there, dark-eyed white, and then MIT is F. And then the bottom there, we've got some other ones. Premature graying. So you have A, which is a couple months old, B is 1 year later, and look at C, 2 years later, all white. All right. Those are all the breeds. The classification used here was based primarily on color and in accordance with the coat color standard and pattern of the American Ferret Association. Coat color is divided into 3 classes: coat with white markings, coat without white markings, and albino. Albino ferrets have solid white hair with red eyes because of deficiency in the pigmentation of skin, eyes, and hair. Coat colors without white marking include sable, pastel, black, and chocolate. Coats with white markings were classified according to the white pattern. All ferrets of this class have white feet. Panda ferrets have an entirely white head. American panda ferrets have large white areas anywhere on the trunk, head, and feet. Blaze ferrets have a white stripe on the head that begins in the muzzle and extends to at least the level of the ears. Dark-eyed white ferrets are solid white, but unlike albino, they have dark colored rather than pink eyes. And then juvenile dark-eyed white ferrets also have darkly pigmented line across their back. Silver ferrets have variable proportion of white hairs throughout the coat. Ferrets with a coat pattern including white feet and a white bib, and sometimes other white patches of white on the ventral portion of the abdomen, stifle, and head were classified as mitt pattern. Some coats with white markings change over time, called premature graying. Therefore, after testing, a follow-up check of coat color was done when possible to see which one of these ferrets had the premature graying trait. Angora ferrets had a long coat, lack of an undercoat, bifid nostrils, which is an extra fold of skin on the nostrils, and small tufts of hair on or inside the nose. In addition to bear testing, they did otoscopic examination as well. Results. Ferrets were from various European lineages: French, Belgian, German, Dutch, and Danish. 71% had normal hearing, 29% deaf. Of the deaf ones, 22% bilateral, 7% unilateral. There was no association between deafness and sex. There was no association between hearing and the Angora trait. There was a significant difference observed in the prevalence of deafness between white-marked ferrets and non-white-marked ferrets. All pandas, American panda and blaze, were deaf. All ferrets without white, including black, chocolate, pastel, and sable, had normal hearing. 31% of mitt ferrets, and then a handful of without other white markings, and then a handful of mitt ferrets with other white markings were deaf. The percentage of deaf ferrets was significantly higher amongst ferrets with premature graying, 87%, than in ferrets without premature graying at 9.6%. All right, discussion. In dogs, over 80 breeds have identified with this deafness disease. It's routinely and objectively assessed by means of bear testing for the breeds at risk. The highest prevalence at 30% of pigment-associated deafness with 8% bilateral and 22% unilateral has been reported in Dalmatians in the United States. In this study, the observed prevalence of deafness was 29% in ferrets, similar to the Dalmatians in the US, but more bilateral at 22% than unilateral at 7%. In this study, they observed no significant association between deafness with sex or Angora trait, but a strong association with coat color. Ferrets with no white markings did not have deafness, whereas panda, American panda, and blaze all were deaf. It is known that the white coat color can be the result of expression of two phenotypic traits: white markings and albinism. Oculocutaneous albinism, caused by lack of pigmentation in the hair, skin, and eyes, and occurs in several animal species, including ferrets. Albino ferrets are homozygous for a mutation identified in the TYR, which is the tyrannase gene. In albino animals, melanocytes are present, but tyrosinase, which is the enzyme that catalyzes 3 steps in melanin synthesis, is inactive. During embryological development, melanoblasts are distributed normally, but persist as clear cells. Albinism is the result of impaired expression or function of pigment-producing gene. By contrast, white spotting results from defects in pigment cell differentiation, proliferation, or migration from the neural crest to skin or hair during development. This results in a lack of melanocytes in the entire skin or in patches. White spotting is therefore the result of impaired expression or function of pigment distribution genes that control whether melanoblasts migrate to the skin and how many melanocytes are contained in the skin. In mammals, several loci are responsible for white markings or a completely white coat. Dominant white, which is a big W locus in cats, piebald or white spotting, which is a big S locus, and mural, which is big M locus in dogs, and Waardenburg syndrome in humans are all associated with congenital deafness. Deafness is caused by degeneration of the stria vascularis of the cochlea in the inner ear. Intermediate cells of the stria vascularis are actually melanocytes that are derived from neural crest. In several mammals with primary neural crest defects, such as white spotting, deafness is associated with the absence of melanocytes in the stria vascularis. Although the exact pathophysiology is unclear, melanocytes, but not melanin, appear to be vital for normal stria vascularis development and function. On the other hand, albino animals are not usually afflicted with deafness because they have a normal distribution of melanocytes in the skin and inner ear. Furthermore, albino phenotype conceals other coat colors and patterns carried, so the albino genotype is epistatic over all other coat color mutations. Now, acquired deafness, like from drug ototoxicosis or otitis media, is also possible. Except for albinism, the genetics of coat color in ferrets is unknown. In dogs with white spotted or piebald phenotype, the S locus has been mapped on chromosome 20, and complex polymorphisms were identified in the vicinity of the MITF gene. Additionally, mutations in the EDN3, which is the endothelin 3, or the EDNRB, which is the endothelin receptor type B, or PAX3, which is paired box 3, IMATF, and SOX10, which is SRI box 10 genes that are involved in cochlear melanocyte development have been associated with white markings and phenotypes and deafness in various species, including dogs, horses, rats, and mice. In humans, mutations in the same genes and in the SNAI2 gene, which is snail homolog 2, cause several types of Waardenburg syndrome, resulting in deafness, blue irises, and a stripe of white in the hair and the beard with premature graying and minor structural facial deformities. Waardenburg syndrome is a consequence of abnormal proliferation, survival, migration, and differentiation of neural crest-derived melanocytes. This syndrome in humans shares similarities with deafness in ferrets, with a white stripe on the head and premature graying. However, abnormalities like facial deformities and other defects of tissues derived from the neural crest known to happen with disorders of melanocyte development have not been identified in ferrets. Other than deafness, other health-related problems like short life expectancy or reproductive disorders could be associated with white markings, according to some owners and breeders. Now, premature graying was strongly associated with deafness, 87% in this study. The graying trait in horses associated with non-ultraviolet induced melanoma and premature graying in mice are related to the expression of the MITF gene, which is crucial for melanocyte development and survival. Hair graying is caused by loss of melanocyte stem cells rather than cytotoxic damage of mature melanocytes in the hair matrix. In ferrets, the American panda coat is consistently associated with premature graying. Unfortunately, this trait may not become evident for 2 or 3 years, during which time the ferret becomes completely white. Without prior knowledge of the color at birth, it may be impossible to characterize prematurely gray ferrets as having dark-eyed white panda, American panda, or other white markings. Moreover, it's impossible to know whether a juvenile white-marked ferret will be affected by premature graying. Some ferrets with premature graying were tested as deaf as young as 4 months old before the graying began, and they were still pigmented. Consequently, deafness in these ferrets does not appear later in life with graying. Another interesting finding is that all ferrets with large amounts of white on the head were deaf. Some ferrets with just small spots on the head, such as mitt ferrets with other white markings, had hearing or were deaf otherwise. This observation has already been made in deaf American paint horse and deaf border collies with a greater amount of excess white on the head than those with hearing. On the other hand, some ferrets with no white markings in the head but some on the body, like mitt ferrets, or with white hair disseminated in the coat, like silver ferrets, were also deaf. As in dogs, unilateral deafness does not seem to pose problematic, except with difficulty in localizing sounds. Bilateral deafness in dogs and cats can be associated with behavior problems like anxiety, aggressiveness, and training difficulties. Owners report abnormal social interactions with other ferrets, biting tendencies, and louder than normal vocalizations. Finally, to reduce the prevalence of deafness in the species, like panda, blaze, American panda, and other deaf ferrets should not be bred, especially together. Breeding only bloodlines without a white pattern seems to guarantee the absence of deafness. However, white patterns are popular with breeders and pet owners. One possible strategy to minimize deafness while maintaining desirable white coat markings would be to breed hearing mitt ferrets. Simple guidelines, such as breeding only bilaterally hearing bear-tested ferrets, avoiding repeated matings that result in the high rates of deaf ferrets, and avoiding bloodlines with a high rate of deafness is recommended. So pretty cool, great study. I do wish they had a larger number of ferrets in each coat category, but the point was effectively sold on this. So I'm not sure I agree with bear testing as being a simple clinical tool. It does require general anesthesia, and so there's always risks with that. So even though these are like young, healthy ferrets, that you're testing with bear testing, I wouldn't call it a straightforward clinical test, and most clinics don't have bear testing. The ones that I've dealt with, I've worked with bear testing before for Cavalier King Charles Spaniels that have Chiari-like malformation, because some of them may have deafness, or at least it tells you if the brain is functioning appropriately outside of like a functional MRI, which we don't really have access to. But otherwise, outside of specialty centers who don't really deal with ferrets anyway, I'm not sure bear testing is as simple as they make it seem. So that part I probably would correct. Or maybe in France it's more commonplace, but I know in the States it's not as common. All right, guys, we'll see you in the next video. Thank you so much.